Assalamu alaikum students. Today we are going to learn about the cell division, that is mitosis. Before we have studied the cell cycle, now we are going to revise the cell cycle along with the mitosis. There are some questions which are usually been asked, that why cells divide and what they have to do to accomplish this. So there are how this division of the prokaryotic cells takes place, how the division of the eukaryotic cells takes place, and what about their chromosome structure and phases of their life cycle, and how do the cell grows, and what are their growth factors which are responsible for their growth. So why divide cell division? Why the cell divides? To ensure the persistence of genome precisely replicates DNA and equally distribute the DNA to opposite end of the cell and separates into the two identical daughter cells. Why they divide? Because to make the genome, what we say, persist or to be present in the next generation. So for this purpose, DNA replicates, DNA makes its own copies, and then the chromosomes goes towards the opposite side of the opposite ends of the cells, and then get separated into the two daughter cells. And a strategy to counter the loss of surface area volume ratio as a growth as it cell the grows larger. So the cell do not grow larger at a single cell, but it will divide into the multiple cells in the smaller size to lower the to counter the loss of surface area volume ratio. So when there is a larger surface area, the volume ratio will be lowest. So that's why we see that this strategy is nature has produced to divide the cells to increase the surface area to volume ratio to as compared to as compared to the cell grows, as a single cell grows larger, so its surface area will be becomes less. And it also permits growth and development of the multicellular organisms. Because of the cell division, the multicellular organisms are being produced, and allows the replacement of the damaged or dead cells, such as genome is the possess the DNA which is unique to the each species. In prokaryotes, the cell divides through the process of binary fission. And in eukaryotes, the cell divides by the mitosis. For example, in human life cycle, you see here, see, this is the individual which has a 46 number of chromosomes. So what they do, if the female is there, it will release the ovum which possesses the 23 number of chromosomes, and the male which release the sperm which also possesses the 23 number of chromosomes, because their gonads produce the number of chromosomes, half number of chromosome cells, which are converted into the sperm and ovum respectively in male and female. So you see here that when they fuse together, they form the zygote, and 23 plus 23 become the 46 number of chromosomes. These 23 came from 23 came from the father, and 23 came from the mother, and become the 46 number of chromosomes. Then here, after the zygote formation, mitotic division takes place, and up to the adult formation. After the adult formation, again, if there is a male adult, then it will produce the sperm. If there is a female adult, it will produce the ovum. So this is the fertilization human life cycle, in which the one part of their life cycle goes through the mitotic division, and other life cycle part goes towards the meiotic division, and fertilization takes place in between them. What about the prokaryotic cell division? They divide by the binary fission, in which the most of the genetic material is incorporated into a single circular chromosome made of the double-stranded DNA and associated proteins. It contains 1 out of 1000 of eukaryotic DNA. It means if it is too much small as compared to the DNA, eukaryotic DNA. If there are this 1000 genes present in the eukaryotic DNA, then only one gene will be present in the prokaryotic DNA. If we say that if the size of the eukaryotic DNA is 1000 nanometer, it means the size of the prokaryotic DNA would be 1 nanometer. This is the difference, size difference between the DNA. It's still highly folded and packed into the cell. Still, this as a small genome, it also is very much highly packed, and it contains very much genes, multiple genes present inside their genome. The binary fission is the chromosome replicates into the identical loops, and each attached to the plasma membrane at the adjacent sites. Between the attachment sites, the membrane grows and separates the two copies of the chromosomes, which are being copied further, and bacterium grows to twice its size and cleavage furrow develops. So now what will happen here? You see that the replication or making the copies of the DNA starts, and after the copies they start detaching from each other, and the detachment before the detachment the cell becomes enlarged and grows to twice of its size, and the cleavage furrow will develop in between the both of the in between the cells to divide the cell into two halves. The cell wall develops across the bacterium between the two chromosomes, and which will cause them to separate in between two others. You see here, this is the it was first in this size, but when they start replicating, they become it becomes bigger in size, and the cleavage furrow is being formed in between the two cells, and the cell is divided into two halves. You see, the many organisms, especially the unicellular organisms, also go for the binary fission. They reproduce by means of the cell division, that is also called the asexual reproduction. Cell division in the eukaryotic cells. Eukaryotes. The chromosomes consist of a DNA proteins complex called the chromatin. Protein include histones that aid in coiling of the nucleic material in dense visible chromatids. Mitosis duplicates chromosomes to pair a sister chromatid. Duplication in is very precise. Only one error in one lakh chromatids can be found. We can see that and since each replication of chromosome poles of the cell, the mitosis may be followed by the cytokinesis. After mitosis, there will be the cytokinesis also takes place. So in cell division, what will happen is that the chromosomes, which consist the DNA and protein, and the company by the combined state of the DNA and protein, they are also called the chromatin material. And that chromatin material consists the histone proteins, which are wrapped by the nucleic acid material that is known as the DNA, and that's why they are looks like as the beads of the DNA chromosomes. Okay, so let's see if before going to this that division, we also want to know that what is that cell cycle. The mitosis is the smallest part of the cell cycle. There are the three other phases which I showed you the last week in our lecture, that these are the three phases: G1, S phase and G2 phase. About 90% of the time of the cell is spent in the interphase stage. What is interphase stage? This complete red color is called interphase stage, in which the G1, the first growth phase, and S, the duplication of the DNA, and G2, the second growth phase occurs, and only the 10% is available for the mitosis, which is used by the process of the cell division, which divides the cell into two equal halves and produces the two daughter nuclei, two daughter cells. Mitosis, division of the nucleus, and cytokinesis takes place in the mitosis process. So, complete cell cycle consists of the four phases in which the mitosis, G1, S phase, and G2 phase. The and if we want to distinguish into two phases, then it means it has the one is the interphase stage, another one is the mitotic phase, which is also called as M phase stage. So in interphase there are subphases that is known as the G1, S phase, and G2 phase. And M phase also possesses the four phases: prophase, metaphase, anaphase, telophase, and the cytokinesis. Okay, so these are the various parts of the cell cycle. And you must remember that this interphase accumulates about the 90% of the cell, 90% of the time by the cell, and only 10% of the time is required by the mitosis. Now, cell cycle. How long is one cell cycle? It depends on the cell. Skin cells usually go for the 24 hours. Nerve cells never after the maturity. Cancer cells very short within the 20 minutes. Remember, every cell only has a certain number of divisions it can undergo, then it dies. Apoptosis is a programmed cell death by which they will die after the number of cell divisions. So it means that cell divides by a limited number of times. It does not divide unlimited number of times, that it will divide and divide and divide unless you dies. But the cell has the number of limited number of time, limited number of times, that how much times it divides. After that times it will die, and then after its death, this process is called the apoptosis, means the programmed cell death. It means that cell is already programmed to die after the two divisions, two times cell divisions, or four times cell divisions, or eight times cell division. It depends upon the uh, type of the cell where they are present. So you must remember this thing, that apoptosis is the process by which the cells divide according to their programmed death. Interface. Now, the interface stage, which we have already studied, there are three phases: G1, S, and G2. In G1, which is also known as growth, first growth phase or first gap phase, in which the small cell is absorbing nutrients and growing and doing its job. And S1, which is also the synthesis phase, in which the cell is continuing to grow and duplicates its DNA. And G2, which is also the second growth phase or the second gap phase, which cell keeps growing and doing its job. Now, G2 phase is characterized by the well-defined nucleus bounded by the nuclear envelope and nucleoli, and these a pair of centrosomes adjacent to the nucleus, which is division occurs in the S phase. In animals, a pair of centrioles in each centrosome. In animals, the in microtubular array ester around each centrosome is found, and duplicate chromosomes cannot be distinguished. Chromatin too loosely packed but cannot be distinguished with each other from each other. So in mitosis, we can have the closer look. The DNA is all twisted up into the chromosomes. You see, this is the DNA which is twisted up into the form of chromosomes along with the proteins. As I told you that the histone proteins are wrapped with the DNA, and it is too much compacted and twisted with each other to form the chromosomes. Okay, during the S phase, chromosome is copied and complete identical sets of chromosomes are being formed, and they are connected in the middle by the centromere. Okay, so this is this part of the centromere from where these two chromosomes are connected with each other. Okay, a single copied chromosome is called the chromatid. Okay, single one. This single copy is called the chromatid. The original cell is called the parent cell, which divide to give rise to new cells are called the daughter cells. Before cell division occurs, the cell replicates and makes its own copies of the DNA. So each daughter cell gets the complete set of genetic information which was previously present in their parent cell. So each daughter cell is exactly like its parent cell and same kind of the number of chromosomes as the original cell. So you see here, this is the parent cell in which the parent cell is producing its copies. Red one produces its own copy and white one also produces its own copies, becomes the homologous chromosomes. These homologous chromosomes are dividing by the process of mitosis and giving rise the two sets, and the two sets, one set is giving the one daughter cell, and other one is the giving the other daughter cells. So this is how you see the parent cell and gives rise the exact copies of their uh, the daughter cells are having the exact copies of their parent cell also. So mitotic phase is divided into the four major stages. Actually, there are two stages. One is the karyokinesis and other one is the cytokinesis. The karyokinesis again divided into the four stages: prophase, metaphase, anaphase, and telophase. While the cytokinesis is the simple one where the cytoplasm divides and gives rise to two daughter cells. In karyokinesis, as I told you, there are four stages that is also called as the PMAT, prophase, P for prophase, M for metaphase, A for anaphase, and T for the telophase. So in four mitosis directed by the four phases, this prophase in which the spindle is formed and nuclear membrane start disappearing and the chromosomes start forming their own copies, and in metaphase they align in the cell plate where the this spindle is attached on the each side of the chromosome with the help of centromere, and that is a protein which is connecting the two homologous chromosomes, and then anaphase cuts apart two uh, uh, chromatids from each other, and then in telophase what will happen that this nuclear membrane start appearing on the both sides, and the nuclear furrow cytoplasm furrow is appeared in the chromosomes in the cell. In prophase, let's start. The prophase, characterized by the disappearance of the nucleoli, chromatin fibers condenses into discrete chromosomes composed of the two identical sister chromatids joined at the centromere. Okay, and formation of the mitotic spindle composed of the microtubules uh, between the two chromosomes takes place, and centromere move apart, migrating across the surface of the nuclear envelope. So you must remember this, that how these chromosomes are taking place here. That in the prophase, what will happen? First of all, you must remember that nucleoli will start disappearing, and second, the chromatin material which was absorbed in the G2 phase can start condensing with each other and forming the thread-like structure and looking like a thread chromosomes now. And the spinal formation takes place with the help of the centrosomes, and then chromosomes start move apart and migrating across the surface of the nuclear membrane envelope. We see here, this is the condensation nuclear membrane nucleolus is going to disappear, the chromosomes coil up and nuclear envelope disappears and spinal fibers are formed. And prometaphase, this is also the part of the prophase in which the nuclear envelope fragments and fragments are dissolved, and also spinal fibers extend from the each pole towards the uh, cell equator, and each chromatid has a specialized center called the kinetochore located in the centromere, and on which the kinetochore microtubules are attached to the kinetochores. Non-kinetochore microtubules form each centrosome overlap uh, each other. So to strengthen the spindle. In metaphase, what will happen that in metaphase centrosomes are at the opposite poles of the uh, cell, and chromosome migrate to the metaphase plate. The chromosomes of all the centromere of all the chromosomes are aligned on the metaphase plate, and kinetochore of the sister chromatids surface face opposite poles, and so that they the identical chromatids are attached to the kinetochore fibers regarding from the radiating from the opposite ends of the parent cell. The entire structure formed by the kinetochore and non-kinetochore fibers is termed the spindle. Let's see. This is the picture in which you see this is the metaphase plate in which these chromosomes are attached with each other, and this part you see in this part is facing the kinetochore, and this is also the kinetochore where the centromere is also present. On the centromere, these kinetochore are attached from this opposite sides, and where these spindles are attached with each other. And this is the metaphase plate, and on the basis of this, these are being attached, and the chromosomes line up on the middle of the cells, and spindle fibers connect to the chromosomes. Okay. So in anaphase, you see the characterized by the sister chromatids split apart into the separate chromosomes moving to opposite ends of the cell. Depolymerization of the microtubules at the kinetochore and takes place. The chromosomes move centromere first, moving the chromatids in a V shape. Okay, and then the poles of the cell move further apart the elongating cells. At the end of anaphase, the two poles of the cell have identical collections of chromosomes. Let's see in the pictorial form. See, this is the V-shaped formation takes place in the chromosomes when they took the chromosomes from each other. And what will happen that you see that these chromosomes are get separated from each poles in the anaphase stage, and these two are very much identical to each other. They are called the sister chromatids from each other, and these are also the sister chromatids. These color shows the sister chromatids, and these move towards their opposite poles. The chromosomes divide, copies divides, and spindle fibers pull chromosomes to opposite poles. Now, the last phase that is known as the telophase stage. This is the characterized by the non-kinetochore microtubule further elongate the cells. The daughter nuclei begin to form at the two poles. The nuclear envelope forms around the chromosomes from fragments to parent cell's nuclear envelope. Nucleoli reappears, the chromatin proteins uncoil, and chromosomes uh, become less distinct. Okay, what will happen here that in look at this picture, and we will see here these chromosomes will when they get separated, they become uncoiled, and they become less visible, and also the n- nuclear membrane start appearing on the both sides of the chromosomes, and also their nucleolus is also appearing on the both sides, and they one cleavage furrow will also be found, which will distinguish or this will disintegrate both daughter cells from each other in the next stage. So chromosomes uncoil, nuclear envelopes forms, and two nuclei are formed in which the spinal fibers will also be disappeared, and centrioles will also disappear from the site. And now the karyokinesis have been completed here. Now the cytokinesis will start. In animal cells, the cleavage furrow forms and pinch off the two daughter cells, and done by the contractile ring of the microtubules on cytoplasm side of membrane. While either in plants, a cell plate, old metaphase plate, grows between the two daughter cells, and the cell membrane grows in either side of along the length of the cell plate, which becomes the cell wall. Now, by the end of the cytokinesis, the two separate daughter cells will, with genetically identical nuclei, were give present. So in this picture, we can see here in the plant and animals, the cytokinesis, the division of the rest of the cell cytoplasm and organelles will take place in the cytokinesis after the nuclear division. In animal cells, the cytoplasm pinch off. You see, this is the from here they pinch off and gives rise in two cells. In plant cell, the cell plate is formed. This is the cell plate, which was originally the metaphase plate. There, here the cell plate will be formed, and the cell membrane will be formed here, and later on, their cell membrane will be converted into the cell wall. After the mitosis and cytokinesis, the cell returns to the interphase stage and to interface to continue to grow and perform the regular cell activities, just like from this G1 S phase and G2 phase. Okay, if we go through the complete life cycle of the what we say mitotic stage, see here, this one is the original one, and you see in with the help of electron microscope we can see his here. This one is a diagrammatic view. This is the nucleus, and this red color shows the nucleoli, and these thread-like structures are called the chromatin, and this is the centrioles. Next stage, what will happen that you see in the blue color shows that chromosomes are start appearing in the nucleus, and the nuclear membrane start disappearing here, and the chromosomes start appearing down from the both sides. You see, and here also this is the nuclear membrane, and these are the chromosomes which are appeared in the nucleus, and they form their own copies. You see there are the two here, and they becomes the four here. They becomes the forms their own copies, and the nuclear membrane start disintegrating. You see it is not now deformed, and they becomes on the opposite pole, and they are going on the metaphase plate. They are make aligning themselves on the metaphase plate. In the complete metaphase plate, you see this, they have aligned themselves in the metaphase plate, and this is the greenish part which shows that they have aligned themselves in the metaphase plate. This is the spindle. Okay, and in the next stage you see this is they are going to separate in the v-shaped. This is the v-shaped structure in the anaphase stage, and this picture also shows the early anaphase, and this is the late anaphase stage, in which you see separating into the chromatid formation. In the next stage, yes, that's great. And you see, this is the nuclear membrane, and in this nuclear membrane, this start appearing here, and this pinch off is also forming. This is the disintegration of the spindle also takes place, and they will loosen their tightness. And now you see there's the chromosome chromatids are at one side, and their sister chromatids are on other sides, and they are forming the again the nuclear membrane and also the nucleoli. You see here they have becomes, and this is the nuclear membrane is forming on the both sides, and then again this complete nuclear cytokinesis takes place, and they have been divided into the two daughter cells, and where the chromatin material and chromosome nucleoli and centrioles are present there. And you see this, these are the in original form. You see these the two daughter forms are cells are being formed, and this is again start growing and forming the uh, yes, they start repeating until they form the tissues or whatever they are where they are present, and their life cycle is limited. And as I told you earlier that they do not divide in multiple times. They divide for the certain number of times which has been given them, and then they die on their programmed death, that is called the apoptosis, which we will discuss in our next lecture. Okay, let's examine in the this complete phase with the help of this tabular form in which the phases and their chromosome appearance and location and the important events took place. In interphase, what will happen? The DNA copies itself in the form of chromatin, and important events takes place: the DNA replication, DNA growth, and cell growth, and replication of the organelles takes place. Which organelles will replicate? The mitochondria and the other organelles such as chloroplast. In prophase stage, the chromosomes coil up again and the nuclear envelope disappears and a spindle fiber forms. In metaphase stage, chromosomes line up in a metaphase plate which is called in the middle of the cells, and the spindle fibers connect the chromosomes with the help of a spindle on the kinetochore which is present on the centromere. And in the anaphase stage, these chromosomes copies are divide and move apart, and the spinal fibers put the chromosome copies apart to the opposite poles. In telophase stage, the chromosomes uncoil and bake into the chromatin, and a nuclear envelopes forms to two nuclei are formed. Spinal fibers disappear. Okay, and the cytokinesis chromatin material, again the chromosomes uncoil themselves into the chromatin material, and then the division of the rest of the cells, cytoplasm and organelles takes place. This is complete what we say uh, summary of the mitosis. What is the control of cell division? What are the cues include? The cue include the growth factors may bind with the membrane receptors to promote the division, and density dependent inhibition affected by the nutrients and space. G1 phase is also responsible as a point of the no return, where the term of the restriction point passes to one of the cell of past which is the which a cell must go into the S phase. So there is some checkpoints, as I told you in previous turn, that by which they have to pass some mm, that qualities to go towards the next stage. If the cell doesn't go past restriction point, the it goes into the G naught phase, in which it will not go for the further division. Cell size, the ratio of the cytoplasm to genome ratio, must be high enough to allow the cell to go for the restriction point. Okay, so these all things must made after, before the cell division. The mitotic clock is still mostly unknown processes. When the mitosis takes place, no one knows, and but no one knows about its process. The onset of the S phase appears to commit the cell to G2 and M phase. The cell cycle events are synchronized by rhythmic changes in regulatory proteins called the protein kinesis that catalyze the phosphorylation of a target protein by ATP. The phosphorylation activates or inhibits the large target protein's activity. The cyclical, what we say, the cyclical changes in kinesis activity are controlled by a second group of regulatory protein called the cyclins. Protein kinases that regulate the cell cycle are called the cyclin-dependent kinases. CDKs are those which regulate the cell cycle. CDK concentration, there are multiple proteins. This is not a single protein, but there are so many proteins which are responsible to regulate the cell cycle. CDK concentration stay the same throughout the cell cycle. However, its activity varies. For example, active MPF, maturation promoting factor, that phosphorylation and chromatin proteins causing the chromosomes to condense and interphase in prophase, and phosphorylation of the nuclear envelope in prometaphase promotes the fragmentation. Cyclin is another type of protein cyclin-dependent kinase produced throughout the cell cycle, accumulates through during the interphase. Cyclin combines with the CDK to make active MPF, through which peaks in concentration with the cell cycle. Within cyclin, MPF initiates the mitosis. At the end of mitosis, MPF activates an enzyme that destroys cyclin. You see here that how these proteins are promoting with each other. You see these are in mitosis, the range of the interphase and the other phases. Cancers that cells lacking the division control. The cancerous cells do not respond to the standard cellular controls, growing and dividing until the nutrients are exhausted. Cells that become cancerous are said to have transformed. Immune system usually destroys such cells, but if not, cancerous cells coalesce tumors. So our immune system is continuously or usually destroys all type of the cells. These all type of the cells occurs inside our body most regularly, and they are being destroyed on the regular basis. But if not, they will become in the tumor cells. If cell remains at the tumor, then the tumor at its beginning, and if the cell is spread out, then tumor is malignant. Spread is called metastasis. The causes the cancerous cells is probably caused by the alteration of the genes that control the cell division. Cancer means I don't, Talha, I don't remember I don't know the name meaning, but I can say that it means that the multiple cell division without any without following any protocol or any losing without any principle. So we are losing our control on these tumor cells or these cancer cells because they are not our under control, and they are continuously dividing. That's why our immune system destroy them, because they are not uh, in our communication. They are out of communication after becoming the cancer cells. Okay, so this is the interphase stage. This is the prophase and metaphase, anaphase, and telophase. This is the uh, actual pictures of the mitosis, in which we can see here that these are the uh, cell divisions or processes, stages of the cell division. What happens after the mitosis? The cell returns to the interphase chromosome. S uncoil backs into the chromatin, and cycle repeats itself over and over. And at what stage are the our cells in these uh, at in the cell cycle? Different cells can be different stages: interphase, mitosis, and cytokinesis. You see this is the skin cells, and these skin cells are at the different stages on the different areas of the cells. They do not all goes for this starting from the cell cycle to end of the cell cycle, but they have the different stages at the different various parts of the cells of the body. The guarantee the products of the mitosis is the two cells. The daughter cells are identical to each other and to the mother cells. uh, why is so important? You see this is the mother cell, and their daughter cells is the so identical to each other. Why it is so important that they should be identical to each other? This is the question arises. In humans, the two daughter cells will have 46 chromosomes, 23 pairs of number of chromosomes, and each chromosome is said to have the same gene sequence. The beauty of asexual reproduction is that the mitosis is the chromosome is a form of asexual reproduction in which the individuals are produced by one parent, and thus the identical bodies or identical organisms are being produced. This is all of the today's lecture. Tala, hope you enjoyed and hope you have learned a lot from here. And if you have any question, you can ask me.